Welcome to Unit 4 Biotechnology, an essential component for CUET PG Life Sciences preparation. In this lecture, we will explore the fundamental principles that form the backbone of modern molecular biology and genetic engineering. Biotechnology is not just a theoretical subject, it is a powerful applied science that drives medical innovation, agriculture productivity, industrial manufacturing, and environmental sustainability. This lecture is structured to help you understand both conceptual frameworks and their practice applications, which are frequently tested in competitive exams. In today's session, we'll cover the core pillars of biotechnology. We will begin with recombinant DNA technology and gene cloning, which forms the technical foundation of genetic manipulation. Then we will examine real-world applications in medicine, industry, and agriculture, highlighting how biotechnology transforms research into sustainable products. Next, we will discuss plant and animal cell culture systems that enables controlled biological production. Finally, we will explore environmental biotechnology, focusing on pollution control and sustainability. The sequence mirrors the concept progression for molecular mechanism to apply technologies. By the end of this lecture, you should be able to explain how recombinant DNA technology works at the molecular level, describe the stepwise procedure for gene cloning, identify key molecular tools such as restriction enzymes and cloning vectors, also evaluate major applications of biotechnology across different sectors. You should also understand the biological basis of plant and animal cell culture, also explain how biotechnology contributes to environmental management. These objectives correspond directly to exam-oriented conceptual manner. Biotechnology refers to the application of living systems or biological components to develop useful products, processes, or technologies. It is inherently interdisciplinary, integrated molecular biology for gene manipulation, genetics for inherited patterns, microbiology for microbial systems, biochemistry for metabolic pathways, and engineering for large-scale production. Its major branches include medical biotechnology for therapeutics, agriculture biotechnology for crop improvement, industrial biotechnology for biochemical production, and environmental biotechnology for ecological management. Essentially, biotechnology converts biological knowledge into practical solutions. We now begin with the most fundamental and transformative concept in modern biotechnology, recombinant DNA technology. This field revolutionized biology by enabling scientists to manipulate genetic material in controlled laboratory conditions. It provides the molecular framework for gene cloning, transgenic organisms, and therapeutic protection. Understanding this session is crucial because it underpins almost every application of biotechnology. Recombinant DNA technology involves combining genetic material from different sources to create a novel DNA molecules that do not naturally exist. This process allows scientists to isolate a specific gene, insert it into another organism, and express it to produce a desired product. Applications include protein synthesis, functional gene analysis, and genetic modification of organisms. The central principle is deliberated and precise manipulation of DNA sequences to achieve defined biological outcomes. Tools of recombinant DNA technology. Successful genetic manipulation requires specialized molecular tools. Restriction enzymes act as precise molecular scissors, cutting DNA at specific sequences. Next, DNA ligase functions as molecular glue, joining DNA fragments. Next, cloning vectors serve as carriers that transport foreign DNA into host cells. Next, host organism replicate and express inserted genes. Next, polymerize synthesized DNA and selectable markers help identify transform cells. These components together form an integrated system for DNA manipulation and expression. Restriction endonucleases are enzymes that recognize specific nucleotide sequences and cleave DNA at those locations. Most commonly used are type 2 restriction enzymes which cut within recognition sites that are typically palindromic. After DNA fragments are generated, they must be permanently joined to form recombinant molecules. DNA ligase catalyzes the formation of phosphodiester bonds between adjacent nucleotides. Sealing breaks in the sugar phosphate backbone. This enzyme ensures structural continuity of recombinant DNA. Without ligase, inserted gene fragments would not remain integrated into vectors, making stable cloning impossible. Cloning vectors are DNA molecules engineered to carry foreign genetic material into host cells. For effective cloning, vectors must contain an origin of replication for autonomous duplication, selectable markers for identification, and multiple cloning sites for insertion of foreign DNA. Various vector systems exist, including plasmids for small inserts, bacteriophage for effective delivery, and artificial chromosomes for large DNA fragments. Vector choice depends on experimental objectives. Host organisms receive recombinant DNA and serve as biological factories for replication and expression. Bacteria such as E. coli are widely used to rapid growth and well-characterized genetics. Yeast provides eukaryotic expression systems, while plant and animal cells enables production of complex proteins. An ideal host must be grown efficiently, maintaining genetic stability, and express the inserted gene at high levels. Having understood the tools, we now move to gene cloning, the practical process of producing multiple identical copies of a specific gene. This is operational application of recombinant DNA technology. Gene cloning is the process of isolating a specific DNA segment and producing numerous identical copies through biological replication. It allows scientists to analyze gene function, produce proteins, and modify genetic traits. The outcome is amplification of a targeted DNA fragment, enabling detailed molecular study of industrial production. Gene cloning follows a sequential workflow. First, DNA is isolated from a source organism. Second, restriction enzymes cut both the DNA and vectors. Third, ligase inserts the gene into the vector. Fourth, the recombinant vector is introduced into host cells. Fifth, transform cells are selected. Finally, the cloned gene is expressed and analyzed. Each step must be executed precisely for successful cloning. DNA isolation. The cloning process begins with extraction of DNA from cells. This involves breaking cell membranes, removing proteins and other contaminants, and purifying nucleic acids. DNA may be genomic, representing the entire genome, or complementary DNA derived from mRNA. High purity is essential because contaminants inhibits enzymatic reactions. Transformation introduces recombinant DNA into host cells. In bacteria, heat shock temporarily disrupts membrane to allow DNA entry. Electroporation uses electric pulses to create transient pores. Microinjection directly delivers DNA into cells, which gene can propel DNA-coated particles into plant tissues. Method selection depend on cell type. After transformation, only a fraction of cells contain recombinant DNA. Selection techniques identify successful clones. Antibiotic resistant markers allows only transformed cells to survive. Blue-white screening distinguishes recombinant from non-recombinant colonies. Reporter genes produce detectable signals. These systems ensure accurate identification of genetically modified cells. Gene libraries are collections of cloned DNA fragments representing an organism's genetic information. A genomic library contains the entire genome, including coding and non-coding regions. A cDNA library contains only expressed genes derived from mRNA. Libraries are essential for gene discovery and sequencing. Polymerase chain reaction is a powerful method for amplifying specific DNA sequences in vitro. The involves repeated cycles of denaturation, primer annealing, and extinction by DNA polymerase. PCR enables rapid production of millions of copies of a target sequence and is widely used in diagnostics and genetic analysis. Now we move from molecular mechanisms to real-world applications. Biotechnology translates genetic knowledge into practical benefits for society. Medical biotechnology has transformed healthcare by enabling production of recombinant proteins such as insulin and growth hormone. It supports vaccine development, molecular diagnostics, and gene therapy. Techniques like PCR and DNA probes allow rapid disease detection, making biotechnology central to modern medicine. Gene therapy treats diseases with introducing functional genes into patients' cells to replace defective ones. Somatic gene therapy affects only treated individuals, while germline therapy affects future generations. It holds promise for treating inherited disorders and certain cancers. Industrial biotechnology uses microorganisms to manufacture products such as enzymes, antibiotics, organic acids, and biofuels. Microbial fermentation allows large-scale, cost-effective production. It represents the biological alternative to chemical manufacturing. Agricultural biotechnology improves crop productivity through genetic engineering. Traits such as pest resistance, herbicide tolerance, and enhanced nutritional content can be introduced. examples include Bt crops and vitamin-enriched golden rice. These innovations improve food security. Next, we examine cell culture technologies, which allows growth of cells under controlled laboratory conditions. Plant cells can be cultivated in artificial media to produce new plants or valuable metabolites. Techniques include callus culture, suspension culture, and tissue culture. Applications include micropropagation and genetic modification. Totipotency is the ability of a single plant cell to regenerate into a whole organism. This property underlies plant tissue culture and enables cloning of plants from small tissue samples. Animal cells can also be grown in vitro. Primary cultures are derived directly from tissues, while cell lines are immortalized populations. They are widely used in vaccine production, drug testing, and cancer research. Bioreactors provide controlled environments for large-scale cell growth. Parameters such as temperature, pH, oxygen, and nutrients are precisely regulated. They are essential for industrial biotechnology. Environmental biotechnology applies biological systems to environmental production. The field uses living organisms to manage pollution, treat waste, and recover resources. It promotes sustainable environment management. Bioremediation uses microorganisms to degrade toxic substances such as oil, heavy metals, and pesticides. It can occur directly at contamination sites or in controlled treatment systems. Wastewater treatment relies on microbial metabolism to break down organic waste. Activated sludge and anaerobic digestion are major processes that produce cleaner water and biogas. Biofertilizers enhance soil fertility through beneficial microbes, while biopesticides control pests biologically. These approaches support sustainable agriculture. Now we discuss safety and emerging developments. Biotechnology raises concerns about GMO release, gene therapy risk, and ecological impacts. Biosafety regulations ensure responsible use. Emerging trends. Future biotechnology includes CRISPR gene editing, synthetic biology, personalized medicine, and bioinformatics. These technologies enable precision genetic control. For exam preparation, prioritized restriction enzymes, cloning vectors, PCR, gene therapy, tissue culture, bioremediation, and industrial fermentation. To summarize, recombinant DNA enables gene manipulation. Cloning amplifies DNA. Biotechnology has broad applications. Cell culture supports protection, and environmental biotechnology promotes sustainability. Test your understanding by defining recombinant DNA, explaining cloning steps, comparing gene libraries, describing plant tissue culture applications, and explaining bioremediation. Structured learning program. This slide introduces a comprehensive preparation program designed to support systematic learning, including lectures, mock tests, and guide practices. Subscribe for free lecture series and exam-oriented content. Thank you for attending this lecture. Continue revising concepts and practicing questions regularly.